Hi friends of the sun, welcome to our reading group happening every Monday, and you can join it on Zoom yourself with the information that's down in the description, ask questions and stuff like that. And yeah, today we're talking with the author of the OMG dataset paper, where there's like some information about metagenomics. We'll talk about what are metagenomic sequences at all, and then how can we train language models on them, and yeah, what are the useful things and stuffs that come out of these metagenomic language models, let's call it that. Okay, let's go. Good, then I would say let us get into your paper, and I have a bunch of questions that I'll try to find fitting spots to interject them, and yeah, then the people in the audience also feel free to raise your hands or to unmute yourselves. Today we are very few people, so maybe just unmute yourselves and ask away. And yeah, anything else? No, I can't think of anything else. Then let's go. Andre, do you quickly want to introduce yourself and get to your paper?
Sounds great. Yeah, thank you. Yeah, so I'm Andre. I'm co-founder and CTO of Tata Bio. And we are a nonprofit research team building open source machine learning models, tools, data sets to accelerate biotech R&D. My background is in machine learning. Previously, I was in the self-driving industry, working on at Waymo, working on like models for planning. And then I switched over to kind of work more in the genomics space. Today we're talking about the OMG dataset, or Open MetaGenomic corpus, for mixed modality genomic language modeling. Just a quick overview of this talk. I'll go over this data set that we're releasing, as well as GLM2, which is a model that we train on this data set, and also go over some results for protein-protein interaction. And finally, I'll just briefly go over some next steps. So our data set is Open MetaGenomic corpus. So what are metagenomes? So metagenomics is essentially where scientists can sample directly from environments without relying on cultivation. So this like greatly increases the number of things that you can sample. So you can basically see like scientists go in all kinds of extreme environments, or even like within like a human like gut environment, for example, and grab a sample and sequence the microbes in that environment, and then just add this, and then you can deposit this data to repositories like JGI's, IMG, or Magnify. What's interesting about this is that you get like a much more diversity in sequence space than you would from like cultivated microbes. You can kind of think of this like in terms of NLP, like this would be kind of like the common crawl, whereas which is really unstructured data, but potentially very rich in diversity. Whereas something like UniProt is much more structured and well labeled, but just has lower diversity. So I think metagenomics does present a huge promise for as a pre-training dataset source, but it does require kind of like careful curation, just like Common Crawl does. Why are these datasets underutilized? So the first is simply that they're just kind of hard to access. Right now, you'd have to go to like like JGI, for example, and get their data through API calls, which can be very slow and sometimes not work. They also require extensive pre-processing. So because it's so unstructured, like you can frequently have like misassembled contigs or poor quality contigs, depending on the sequencing technology used and assembly method used. And finally, they're difficult to deduplicate and balance. So kind of unlike protein-only data sets where you can run something like MMSeqs to cluster and deduplicate, this is much harder for metagenomic samples because you basically have to align kind of arbitrary region and lengths, and that does get very computationally costly. And also you don't have any taxonomic labels to kind of potentially balance through taxonomy classes. So we're basically presenting the OMG dataset. So it's the largest metagenomic dataset to date. And we combine the two largest metagenomic repositories, JGI's IMG and EMBL's Magnify. And we pre-process it into a mixed modality format for multi-gene modeling. And this is kind of our design choice to essentially make training on this data more efficient instead of raw DNA. So essentially the protein coding sequences are translated into amino acids, and then the intergenic sequences are represented as nucleotides. And then we basically have long contigs with genes followed by intergenic sequences interspersed. And we have quality filtered this data to remove kind of poor assemblies or poor quality data, and we make it easily accessible on Hugging Face. So to visualize this data set, we also release a data set called OG, the Open Genome, which is a subset of OMG that contains taxonomic labels and consists of full genomes. So what we can do is, what we did here is we visualized through UMAP the full data set, or actually this is a subset of the data set. And we embed the data set and display using UMAP. And then we can kind of overlay the colors of the taxonomies for the Open Genome data set, which we have. And then on the right, we show actually the overlay of the Open MetaGenome data set versus Open Genome. And visually, you can see significantly higher diversity, which looks promising.
So where are humans here?
So most of our data, I think like 90% plus, is bacterial and archaeal. There are some eukaryotic samples, but eukaryotic fragments that come from this data, but most of it is bacterial.
Okay. Then where's human gut stuff here?
So human gut can be all kinds of stuff. I'm not an expert, but I think probably like E. coli does get in there too. So yeah, I guess your gut hopefully has a large diversity of different microbes. So yeah, I'm not sure exactly which taxonomies, but the goal is to basically have as much diversity as possible. And you do see kind of clusters that are very densely sampled, which can be a problem with if you try to directly train on this, because you're going to be seeing the same types of bacteria over and over again. So just going over a few like key statistics. So we release. So here I go over OMG and OMG Pro 50. So these are both metagenomic data sets. OMG has 3.3.
Okay, sorry. Let me interrupt you a little bit. Go back a little bit. So what are metagenomes? So you're talking about these contigs. You have this gene calling and so on. Like, what is the raw data that we have, and then how do we know that something is a gene, something isn't a gene, and so on? Like, so maybe first to start off with, basically, how does metagenomic sequencing work, and what comes out of the experiment in terms of data?
Sure, yeah, I can actually, maybe I'll skip forward a little bit to this diagram here. So this is kind of the raw, going from raw data all the way to our like embeddings, but we can focus on this first part to answer your question. So a metagenomic contig essentially is a stretch of data that you have sequenced from a sample. So for example, you grab a sample from the bottom of the ocean, you get a bunch of bacteria mixed in there, and then you sequence the sample to get a bunch of fragments. And then this can be represented as just raw DNA.
So, and then these fragments, are they usually the whole genome of a bacterium, or does it depend on how large they are? Does it contain everything of the bacterial genome?
So almost always it's fragments. I think it's very rare to get full genome assemblies. I think you can think of it kind of just like it's just a mixed bag of a bunch of DNA. And you have all kinds of, that comes from all kinds of different microbes within that sample that are all present there. And then you go through assembly to kind of build fragments that from this data. So you'll get kind of like short sequences that contain up to from just like a few proteins to like thousands of proteins. There's a huge kind of variance there.
Okay, and a few
On the
protein versus 1,000 proteins, on how many um, amino acids would that, or how many nucleotides would that correspond to if we also have these non-coding regions between OS?
Mhm. That's a good question. So, I guess so. If you have a few proteins, proteins are kind of on average length like 250 amino acids. So in our data set, we filter for um, no contigs that have less than 4 proteins. And the reason that is to kind of during training, what we want is the model to learn inter-protein um, uh,
Mm.
representation. So if you think of 4 proteins on average, 250 amino acids, and then the intergenic sequences can also vary, but for bacteria and archaea, they're generally quite short. Maybe at most, like I'm not sure the average, but probably around like 100 or less base pairs. That's kind of the
Wow.
scale. Yeah.
Okay, so and then I suppose we have all these. Okay, so we take our soup, we sequence it, and then we get a bunch of protein snippets, which maybe even are from different bacteria. And then we take these snippets and see that, oh, sometimes these are completely the same, or these two parts, uh, like two overlapping parts and two fragments are completely the same. And then we put them together, and we do that for all the stuff we have in there, and then we end up with such a contig. Is that how it works?
Yeah. So you end up with many contigs. So yeah, you have the soup. You do the process you mentioned, which is you find like the overlapping regions, assemble it, and then you end up with like many fragments from your sample.
Okay, and then these contigs, they could all come from different organisms as well, right?
Yeah. So one contig, comes from one organism, but then if you have thousands of different organisms that are
Yeah.
present in a sample, you'll have many. Um, yeah.
And now you're already saying one contig comes from one organism, right? But if two organisms are very similar, then can't it, for example, have my fragment from organism A and then my fragment from organism B, which has some overlap because that part is the same, and then I have fragment from organism A again, and this part of it is the same again, and then like I have a contig that contains stuff from both
Mhm.
organism A and from organism B. But, like, is there a lot of noise like this, or does this basically never happen?
That's a good question. I'm not exactly sure. I think, yeah, I think it's possible. It. I think it's difficult to, yeah, get like one long stretch from one organism, and I don't know exactly. I think there's techniques to kind of, yeah, minimize uh the like misassemblies between different organisms.
Yeah.
So then you end up with this long stretch of DNA, and then you can call like a gene caller, which will basically find the genes that exist in this stretch of DNA. And
Yeah,
you'll have some genes going forward and some genes going in the reverse direction. Um,
that
and
was
then
also a big mystery. What is this gene calling? How does it, like, basically gene calling, right, is now just a method of you have your contig, which is just a sequence that we think in this order appears in the genome of some organism.
Mhm.
And then how do you find out which organism does it belong to? And then how do you find out which parts are genes? So the second part is this gene calling, but how does it work?
Yeah, that's a good question. There are softwares like Protocol that are designed to do this, and essentially they look for kind of signals in the DNA that signify basically the beginning or end of a protein. So like a start codon and a stop codon, for example. And these can vary depending on the organism type. So there's different like modes you can run these gene callers on. So if you know you're running it on like E. coli, then you can use E. coli. And if in our case, because we don't know what the type of organism is, you can call it in like a meta mode, which essentially kind of will make its best guess of what proteins are, given it doesn't know the exact organism.
Okay.
This process can be noisy as well. So for example, you could end up with miscalled proteins, which might extend much longer than you would expect, or it maybe proteins are not detected and you end up with a super long intergenic sequence. So these are kind of filters that we put into place when curating this data set. Like if there's stretches that are, way too long or way too short, like we will filter those out to kind of avoid these kind of errors.
Okay, and then why or where does this different order or different direction come in?
So this just comes from the fact that the DNA goes and can be read in both directions.
Yeah, but if I assemble this stuff, why isn't it all the same direction?
So the DNA has like base pairs, two strands that are linked. So you can read the DNA going from one direction, or you can read the DNA from the other direction. So some proteins, you'll see the start and the stop codon going from this direction, and then you might also see a protein going from this direction to going this way. And they can even overlap. So you can have like this is how like bacteria like code for proteins very densely, is you can actually have the same stretch of DNA, but that can actually code for two different proteins overlapping going in opposite directions. Um,
Oh, wait.
yes, it's a
Okay.
very compact, efficient way of representing proteins.
I see. Okay, so DNA isn't always read in the same direction.
Exactly, yeah. You can read it in both directions. Yeah, it's much more complicated than natural language.
Okay. So, hmm. We have the like the basic then the complement of the thing is okay, but then what so um each gene has a direction in which it is read, but the
Mhm.
method or the what are the regions in between called the non-coding regions?
Non-coding regions or intergenic regions.
Intergenic. So the intergenic regions, they basically don't have a direction because, right, you don't need a direction in which you read out the gene from the
Mhm.
sequence. And so I have my forward and my reverse, and the reverse, if it's the reverse direction, then it's the complement of the forward that's being read and turned into a protein.
Exactly.
Okay. No, this is weird, interesting. And then, so how often do overlaps like this happen? Because it sounds very, very unlikely that you have a sequence that works as a protein, and then the reverse, like the exact reverse complement of the same thing also folds up into a good
Mhm.
protein.
I think it does happen quite frequently, and I think the reason is just like bacteria are optimized over billions of years to express their proteins and without requiring with minimal energy, and I think that does lead to kind of these optimizations, and they might just be partially overlapping, um, but it's kind of evolved to be read like that.
Anush, what comment do you have on that?
You cannot have full overlap of the same protein read in the forward and the backward because the RNAs would form a double helix. And fully.
Wait a second. You cut out there. You said because the RNA form a double helix and
Wait.
then you stopped. We still can't hear you.
Huh?
It's breaking up. It's breaking up.
I just took out my iPod. So you cannot have full overlap because the mRNA of one gene in one direction would interact with the mRNA of this if you would have the same gene in the other direction.
Okay, so how much overlap can we have?
Well, that is yet to be defined, but
Okay.
full overlap is not ideal for the cell. It's true that they try to have, they like to optimize the space and have genes in both directions, but how much it overlaps, we don't know yet.
Okay, but so do we know that there is some overlap also of the genes?
Yeah, there is slight overlap always, but it tends to be most of the overlappings happen in the same direction, and when there is some in the other direction is small.
I see. Okay. Yeah, the same direction I knew, but the reverse direction I didn't know. And
Then
but
the directionality of the non-coding regions, that is, they are generally considered non-directional, but promoters, like these regions contain information of promoters and ribosome binding sites and all of that. Like for instance, promoters determine the binding of a protein, right? So that
yeah,
needs to have certain directionality because you'll need to read the genes downstream of that. So yeah.
that makes sense. Ben, is it if you also want to say something, just go ahead.
Yeah. Okay, great. Thanks. Yeah, I was wondering in terms of the clustering, I was trying to look through the paper to try to understand when you were clustering these in the sequencing fragments, are you clustering based on the full DNA sequence, or are you clustering the protein sequence? Or are you clustering? How are you clustering?
Right. So that's a great question. We cluster examples, and we just define examples as our pre-processed training set. So we pre-process it to a fixed token length. So in this case, 2048 tokens, and that can represent like around like 3 to 5 on average like proteins. And then we now have like millions of these basically like chunks of the data set. Those are the things that we cluster.
Okay, so just to make sure I understand, are you clustering because you're embedding both the DNA sequence and the protein sequence of these contigs, right? And so the clustering is based on both of those embeddings of those shared embeddings of those sequences, or?
Yeah, so we can actually like, for example, here, this would be one example. So we have our sequence that's pre-processed into proteins and DNA sequences. And then we take this whole thing, embed it, and then average the embeddings over this sequence. And that represents one point.
Okay, great. And you're doing this like basically per contig.
Yes, although contigs, when we pre-process the data set, a contig can turn into like multiple examples. If it's long, for example.
Okay. So you probably answered this already. Are you chopping the contigs into being more or less equal lengths?
Now, Ben, let's get to that later. I would
Okay.
talk about the data, what it looks like, and then we can get to that stuff. Yeah, so we now have our path through the stuff. We know how we assemble our contigs, and then the contigs have multiple genes, and then this gene calling works by looking at start and stop codons, and this way we find out what are the genes. And now you show some statistics of them here. Let's go through them.
Great. So we release actually 3 data sets, OMG, the Open MetaGenomic dataset, OG, which I mentioned here, that's a subset of with taxonomic labels, and then OMG Pro 50, which is a protein-only data set that's clustered at 50% sequence identity. So we have around 3.3 billion proteins and 2.8 intergenic sequences in OMG, which represents in total 3.1 trillion base pairs. For OMG Pro50, this gets clustered into 207 million proteins or centroids, which if you compare to UniRef50 is about 3 times larger. So this kind of signifies there's significantly more diversity in this data set. And then we also show some kind of statistics of the average length of these elements. So previously you're asking about how long were these contigs or scaffolds, and they can be range from, you know, like 10 elements to all the way to, you know, 50 plus elements. There's a large range. So yeah, now I'll move on to the GLM part of this talk.
Okay, 1 second, right? You have this, or in your data set, you have the DNA sequence or the intergenic sequences as base pairs, and the gene coding regions you have as amino acids. So do I also somehow have access to the amino acids? Sorry, to the base pairs of the proteins?
In the current version of the dataset, no, we didn't release that. I think we could in the future.
I find it a bad decision to release it as amino acids. The mapping from like I can just have a dictionary in my code, right, to turn it from base pairs to amino acids, but the other way around I can't do it. So I would just store it as base pairs.
Mm-hmm. Yeah, that's a great point. Yeah, we basically do lose information going that direction. I think the main kind of reasoning is we kind of want the data set to be as easy to use as possible. So if we just release the raw DNA, then users would have to basically run gene calling on the whole thing and do many pre-processing steps. However, yeah, I do see your point that yeah, we should also release the raw DNA, and this is something we can work on.
No, so the way I would use the stuff, for example, is like if you give me the raw DNA, then I don't know what to do with it. But if you give me the nucleotide sequence, then I think it's pretty easy for everyone to understand. Oh, you have here this additional dictionary. If you take these 3 letters, then it turns into this 1 letter. And that's the mapping from DNA to amino acids.
Yes. I think so. Yeah, I guess if we supply the raw amino acids, like because there's a multiple, it's not a one-to-one mapping. We'd have to, yeah, I guess it might require a little bit more thought in how to easily give both of that information to the user.
Could you give the coordinates of the genes for each contig?
Yes, that's also possible, and I think that actually is in the metadata. So when we provide the proteins, they come with an ID, which does include the start and end locations in the contig. So that information is there.
Okay, but right, you said it's not a one-to-one mapping. Sure, but if it's a many-to-one mapping, then it's still nice. Then you store the many thing. You just provide the map. But if it's not a map at all, then that's a problem, right? If it's sometimes in some proteins or in some bacteria, these 3 nucleotides turn into this amino acid, and these other 3 nucleotides turn into the other, and then in the other bacterium, the same 3 nucleotides turn into another amino acid, then yeah, it's not a map. So which is the case? Does it sometimes happen that the same 3 nucleotides encode a different amino acid?
No, I think that's a fixed mapping, I believe.
Yeah, okay, well then I think you should just store the nucleotides.
No, it's not quite true. It's not a 100% fixed mapping. There can be changes, especially when it comes for stop codons and so on.
Okay.
Yeah, I guess there's in biology, there's always exceptions. So there's never a hard rule.
Yeah, there are in fact like many mutation in the nucleotide that can lead to the same amino acid. I mean, you can easily check on Google Image to find something, but like it's roughly 3 to 4 like two to 4 different nucleotides lead to
No,
the same
Dominic,
amino
that's
acid.
not the point. The question
Yeah.
is the other way around, right? We know that many codons map to the same amino acid. But the question is, does the same codon sometimes map to amino acid 1 and sometimes to amino acid 2, depending on which protein it is in? And apparently that also happens. So it's not a map at all.
Yes, I think that's right. There's like depending on the type of organism, they can have a different like decoding table or translation table. And that kind of changes the mapping. So that's kind of all this information is kind of built into the gene caller software.
Yeah. Thank you, Anush. In Anush's thumbs up, so we trust. Good. Then yeah, we have this ugliness that's going on, and probably like what I would find the nicest is if I had just both the nucleotides and the protein sequence. And I mean, then you have like the OMG prot 50 is then 2 times bigger. But at least the OMG or the OMG data set could be like one unified thing, right? I can train my DNA language model on it. I can train my codon language model on it. And I don't have to mess around with amino acids if I don't want to. And like, it's just simpler. And then the OMG prot 50, if I want to do something, like if I want to only train on amino acids, yeah, then I can do that because you provide the amino acids. If I want to do like some amino acids, codon mixed stuff, or like I would say the most niche thing of all the cases is some codon and amino acid niche mixed stuff, and that's right now the thing that you provide. Okay, but all right, you also have just OMG prot 50 where you only have amino acids in there, and I guess that's also a pretty standard thing that we that one would need.
Yeah, I think that's right. I agree we should definitely release the DNA version of the data set. And yeah, I guess it is kind of like a new way of representing this data. Like I guess other pre-trained data sets do not release in this format. I think that maybe the reason we did is because like when biologists look at this data, if you're looking at a raw genome, like you're really looking at a gene map that basically includes this data, which is you have your genes called and then with their directions and positions followed by their intergenic. So yeah, it's niche, but it's also maybe potentially you could think of it as like more natural for the way like biologists look at this data.
Like they're probably, if someone wants to work with DNA, then they would also want to know what the nucleotides are, and not have this information lost. So, I would venture to guess.
Yeah, sounds good. Okay, so yeah, I'll briefly go over the GLM model that we trained. So we train it as a mixed modality language model. So we can tokenize both the amino acids as single tokens and the intergenic sequence. We use byte pair encoding tokens to kind of compress those sequences more. And we train it with standard masked language modeling. So we take our sequence of tokens, mask a random percent of them, and then ask the language model to reconstruct those masked positions. And finally, we train on the semantically deduplicated version of OMG dataset, which I'll talk about in the next slide. And we train at 2 scales, 150 and 650 million parameters.
Just a brief one here. do you concatenate the two protein sequences then simply? If you have now here multiple genes called with an overlap?
Yeah, so in the case of overlap, we just kind of we concatenate them. So
do you indicate this somehow as a special event going on there?
There's no, so we don't indicate that to the model. We just provide the direction.
Yeah,
and
I'm just wondering, I mean, I'm asking because, I mean, after all, there is sometimes a relationship between the distance of some promoter region and the protein that is then expressed afterwards. And now looking at the second protein that's encoded here, you artificially increase the distance between the two. So I'm just wondering whether it might make sense to add another token there indicating that, look, here's something where we chop things together.
Yeah, that's a great suggestion. Yeah, that's true. We're kind of taking this double-stranded sequence and then flattening it into a single thing we can feed to the language model. And by doing so, we do lose this information. And yeah, we can, I think that's a great suggestion to kind of add like position, position encoding maybe that maps directly to the location in the original sequence.
Okay, thanks.
So yeah, I'll talk a little bit about SEMD-dupe. So this is a technique that was released in 2023 from a team at Meta. And the idea is basically if you have a unstructured pre-training data set, can you improve training efficiency by removing near duplicates? Yes.
Okay, it's like, so here the SEMD-dupe, you train your model a bit, right? And then you get embeddings and then you cluster based on those and then you remove, and then you only take some cluster representatives. Is that the deal?
Yes, except you don't take the cluster representatives. You literally compute the distance between like pairs of points in a cluster and then remove ones that are within a certain threshold.
Then we have the question from Ben asking like also about this sort of stuff and how it impacts clustering or not, then maybe not clustering if we want to call that. So when you chop up contigs, did you break them up into discrete or overlapping fragments? So when does chopping up contigs happen and how does it work?
Yes, we chop it up without overlapping fragments.
Why do you chop it up?
Because you need fixed size like input length.
Okay, so your model always has fixed size inputs.
Exactly. I think that's for most, like for example, if you're training BERT, like most people do like 1024 tokens, and that can map to like a varying number of words depending on the tokenization scheme. But generally for training, yeah, you want to train on fixed length sequences.
Okay, and then so now it's kind of you have right, you also have this mixed stuff. So you have a token for, or your vocabulary are basically 24 possibilities where we have 20 amino acids and 4 nucleotides. Is that the case?
Oh, sorry, could you repeat that?
Because how does the mixed modality stuff work? Do you have 24 tokens instead of 20, and then you just have 20 amino acids and 4 nucleotides?
So we have basically all the amino acid tokens as individual tokens. So like you have 24 amino acids, and then we augment that with around 4,000 additional tokens that are generated from BPE, byte pair encoding of the DNA tokens.
Right. I read that. So why do you choose byte pair encoding instead of, like, why don't you do byte pair encoding for both? And why don't you do a byte pair encoding for both?
Yeah, so I think for protein language models, people generally stick to single amino acid tokenization because you're really interested in, I think that helps the model learn kind of like coevolution of individual amino acids, which then correlates with structure. So I think you could potentially train with byte pair encoding, of the proteins, but I don't think that's really been studied. And yeah, I guess also because of the larger vocabulary size with amino acids, you might not actually get that much compression with byte pair encoding.
Okay. I mean, yeah, it also works for language, right? Natural language. And there are models like ProtGPT or so that do byte pair encoding also for proteins. But we have a protein language model person who just turned on their camera. Michael, did you want to comment anything here?
Actually, I wanted to ask another question now that we're back at this slide, but I would also, I mean, I would also always just stick to this single token representation for amino acids. After all, you have many tasks that rely on exactly those embeddings. So I think you
Okay.
just make your own life much easier when you stick to this one token equals one amino acid thingy.
But then is it basically that we don't care about the rest of the sequence? Like, why don't we, or there's no semantic meaning that we care about associated with the rest of the sequence, and that's why there we do byte pair encoding?
Yeah, I think I think in an ideal world, like, yeah, we could probably have single nucleotide and single amino acid representation. This was just our design choice to kind of pack more information into a single example.
Would it be possible to have like a codon representation?
Yeah, I guess that would give you
the sequence is always divisible by 3. It still makes sense also for the intergenic regions to always talk about triplets.
I think for intergenic, you don't have codons for that, so it's not necessarily like, yeah, that might feel
right, just instead of byte pair encoding, which is then something arbitrary, like 5 tokens, you could also just always chop it by 3.
Yeah, but what if I have my gene A and gene B, and the region in between are 137 nucleotides? Then this is not divisible by 3, and then you can't do your triplet encoding. So can that happen, or is it always divisible by 3?
I think for intergenic, it's not always divisible by 3.
Okay.
And I think they're like, for example, the nucleotide transformer models series, those do this kind of, yeah, fixed window tokenization, but they only do it for DNA. So you can just grab some arbitrary sequence and, yeah, just take a chunk that's divisible by 3.
Okay. Well, then that sort of explains to me why we do byte pair for one and not for the other. Okay. Then Michael, you wanted to ask a question.
Yeah, just I mean, after all, here on the slide, you're mixing the both directions, and I was now just wondering whether you make the model's life unnecessarily hard by doing so, so that you could also just essentially show it in one shot, in one forward pass, the redirection A, and then in the other forward pass, redirection B. Because after all, depending on the directionality, you might get different motifs and these things. So, just did you try this?
You also have tokens, right? So what about the direction tokens?
Right. Yeah, that's a great point. So yes, to answer that last question, yeah, we prepend every gene with a direction token, so that either like plus or minus strands. So that does give the model at least that input of which direction. Yeah, in terms of like separating out into 2 separate passes, I think, yeah, that's an interesting idea.
Because after all, I mean, the model would internally first like back translate and produce this like complement strand, right? And then it would only be able to detect the motif. I mean, it would have to do this like implicitly.
Right. Yeah, that's true. And I guess maybe the problem is if you are representing it as amino acids, like the reverse complement is probably different amino acids. So it might be difficult for the model to kind of implicitly figure that out, but yeah, maybe it's possible. I think one benefit of kind of feeding both directions in is like the model then you kind of do get the output representation that includes both strands, and maybe there are interactions between these 2 strands. So that's one benefit.
Yeah, no, I absolutely see the point. I was just wondering whether you did some ablation study on this and just saw then, okay, this works equally fine, and we have exactly this benefit that you just referred to, so we just stick to it. But you didn't try it, right?
No, we didn't try it.
Okay.
I think it would be interesting to see, like, with DNA only models, kind of what it can implicitly learn. Yeah, I think that would be an interesting place to look.
Like, to be honest, sounds to me like the cleanest solution to all of this. You just take your DNA sequence, and then the model is supposed to learn what is a gene and what is not, instead of having your gene caller imposed definitions.
It's not that trivial to know what is a gene and what is not a gene.
Well, then even more so, you should just let the model learn it instead of having your wrong annotations.
But you need to teach it somehow how to learn that, right? You cannot just give a chunk of genome and say, hey, learn what is a protein here.
This is the basic assumption, right? That these models learn to extract such information from raw data. I mean, this is, I would assume that this is just one of the things that the model should pick up first, because there's lots of structure information in there.
The model will learn what are the nucleotides, like how maybe nucleotides follow each other, but it will not learn what is a gene within this DNA.
I would just assume that if you then look at the attention matrix, for example, that you have a lot of more attention going on within a protein coding gene than within an intergenic region. So I would assume that from these patterns, you could deduce where a protein coding region actually is.
Yeah, I
Fingers
think
crossed on that.
Yeah, I think. Go ahead.
Yeah, I also have another question because I am confused about what is the directionality that you give for the proteins, because you are mixing here nucleotides and proteins, right? So then if your protein is read in this direction, you have the amino acid sequence, right? Like that. But then you have nucleotides read from the 5' from the DNA level, but then imagine you have your protein now going in the other direction. How do you put it? You see what I mean? Like, is your protein reversed, or is your protein?
The protein. Oh, sorry. Yeah, the protein is always represented in, like, the direction it's read.
Well,
but
that is probably a big problem. Because then the model is mixing what is the nucleotides that are directly linked upstream of the protein region.
Yes, yeah, that's correct. Yeah, I think, yeah, these are that's a valid point. I think you do kind of run into these complications when representing it in this mixed modalities multi-direction sequence. And yeah, I guess to go back to Hannes's point, I think, yeah, representing as raw DNA is definitely the simplest, like, yeah, no heuristics kind of method. And
okay.
Yeah.
So my proposal of how to do the thing, we have byte pair encoding. No. So yeah, we have an encoding or a byte pair encoding that restricts whenever we have coding regions, the encoding to be our codons. So we have codons for the coding regions and byte pair encoding for the rest of the stuff. And then we add positional encodings, like 2 tracks of positional encodings, where the one or like 3 tracks of positional encoding. One is your standard positional encoding, then the other positional encoding is for the forward direction, and the other positional encoding is for the reverse direction. And in these tracks of positional encoding, you encode, where does a gene start in this direction, where does a gene end in this direction, and then for the other direction, where do genes start and where do genes end. And if you have some other information like this that's similar to genes starting or ending, you can also put that into this positional encoding.
Yeah, I think that's a good idea. Yeah, basically representing the full both strands in full using position encoding. I think that's a good idea. Let's see. Yeah, so I'll just quickly jump back to this slide. So just to explain this figure, so we're looking at semantic deduplication on the OG dataset just for visualization purposes. So we can see the clustering by taxonomy followed by the pruned points. So everything in blue here gets removed from the dataset. And as a sanity check, we essentially see that these pruned points occur in specifically in overrepresented taxa, like Pseudomonodonta, for example, is like the most common one in our data set. And basically, sorry, just another view of this is looking at the distribution before and after. And in the paper, we can compute the entropy of the distributions to just make sure that it increases after deduplication. And the whole goal here is just to improve the balance and improve training efficiency. So in terms of evaluation, the first evaluation we do is using the DGEB benchmark, which is a benchmark we released earlier this year. And just to give a brief overview of what it is, it's designed to be an embedding-based benchmark that covers all branches of life and across multiple tasks. So you can kind of think of this as an analog to MTEB for natural language, which evaluates natural language embeddings on across many tasks like retrieval, translation, etc., and across many languages. So we're basically trying to get kind of the best we can do at holistic view of what these models have learned. So in terms of results, we compared to the ESM2 series. And the results are for the equal number of training flops, we see an improvement in the overall DGB score, which is essentially just averaged over all the tasks. And there is a slight improvement from before pruning and after pruning the data set. And when we look closer, we actually see that most consistently there's an improvement in kind of underrepresented taxa, like, for example, archaeal tasks have a consistent improvement. Sorry, are there any questions here?
No, not really.
Okay. Okay, and then I'll move on to the next part for PPI prediction. So we kind of asked ourselves, like, what is this model learning and is able to pick up on co-evolutionary signal between proteins. So this is kind of inspired by work with PLMs that have been shown to learn within protein co-evolutionary signal. So during training, they learn the mapping between if one amino acid changes in this location, does something else change in a different location? And that can frequently signify some kind of interaction between those two locations or binding, for example. And this can be extracted with the categorical Jacobian, which is from Sergey's group. And the problem is currently the existing tools for PPI prediction are quite limited, and usually they rely on some form of supervision, maybe from like model organisms like E. coli. But it's really hard to expand this to a unsupervised setting over any kind of organism. So we first looked at this MOD-AC protein complex. It's a transporter, and it has been shown also from Sergey's group that there is this co-evolutionary signal between these two proteins. And we run the categorical Jacobian for this MOD-AC sequence over a few models. So first, we run it on the POTS model, which is kind of our ground truth based on MSA. And then we compare it to ESM and EVO. So EVO here is a DNA-only model, and ESM2 is a protein language model, but we still feed in essentially both of these proteins into the context. And then GLM2 naturally takes in both proteins as well. And we see that, yeah, the only signal comes from GLM, and these other models don't pick up on this. And I think it's understandable that ESM doesn't, because it's not trained on sequences of proteins. But it's also interesting to see that Evo here doesn't pick up on it, even though it has basically been trained on stretches of DNA that do encode multiple proteins.
And have you checked this for multiple proteins?
Yeah, so here's another example for toxin and antitoxin. Yeah, and you also see this kind of expected interaction between them. So yeah, so we're pretty excited about these results, and it'd be really interesting to kind of run this at a larger scale over, full genomes, for example, which also might be useful to have like a longer context model that has more than a few proteins.
Yeah.
Okay. And then just to conclude in terms of next steps, so yeah, we're definitely looking into continued releases of the OMG dataset as more data becomes available, and also yeah, based on your feedback, I think we'll definitely try to get the DNA only dataset out as well.
How about both datasets?
Well, yes, yes,
Sorry.
we'll have like the mapping. So yeah, every contig should have both the mixed modality and the raw DNA. For GLM2, we're looking at increasing the context length to get more proteins in the context window, as well as just continuing scaling up the model. And then there's also some interesting directions in terms of generative modeling on the OMG dataset, and also improved dataset balancing. I think there's lots of other ways we can kind of go about balancing this data. Yeah, we're definitely interested in looking into this. Yeah, and also we're hiring. So if you're interested in working or collaborating with us, yeah, feel free to reach out.
Okay, then thank you, Andre. You perfectly made it to the hour mark. And before we get to our after hours and take some more questions, first a big thanks to you for the talk and for answering all of our questions, and also a big thanks to Anush, Michael, and some others who helped us out answering some questions and also asking good questions themselves. But then let's have some more discussion and some more questions. So yeah, and people, if you want to ask something, unmute yourselves, and otherwise I'll go a little bit through the chat and also ask stuff myself. Michael, you are
yeah, I mean, in the meantime, then you can just parse the chat. I wondered about this clustering. I mean, I understand that it's harder to cluster nucleotide sequences than it is to cluster protein sequences, but after all, you have always some code, or most of the time, I would assume that you have some coding regions within those fragments that you process. Did you maybe also compare to what would happen if you just compute sequence similarity based on those coding regions and cluster based on this, just to get an intuition whether this embedding-based clustering is actually going in the right direction, or whether it remains to be better to just do it on the amino acids? So
yeah, that's a yeah, that's an interesting idea. We didn't do that. I guess like alignment over multiple proteins that occur in.
Yeah, I mean, you could also just do it for single ones and then average some quick hack, essentially just to get an intuition whether there is some other way to do it, or whether the embedding based stuff is actually really beneficial compared to some other clustering. Yeah,
that's a good question. So we didn't run too many baselines. I think one we kind of internally experimented with was just balancing based on taxonomy for specifically the OG data. And what we found challenging is like doing just a uniform sampling over taxonomy isn't necessarily like the right decision, because we're arbitrarily assigning this based on our human understanding of these, and some taxonomies have much larger diversity than others. So you can do some balancing there. We found that it didn't perform as well on downstream tasks. So as maybe that as a naive approach didn't work as well. But no, I think it would also be really interesting to look at more like purely sequence-based baselines to see what can we get out of this, because I think the embedding-based deduplication is nice because there aren't really any hyperparameters. You kind of just but there's one hyperparameter, whereas but you don't really have much interpretability besides visualizing on UMAP.
Exactly. This is why I'm also wondering about what the exact effect is. I mean, after all, I mean, for NLP, this is usually much harder because it's like insanely hard to align like human language. But I mean, for nucleotides, there would also be solutions. So you could also probably just align on nucleotide level and see what happens then, right? I thought MM6 also offers this, if I'm not mistaken. Never did it.
Yeah, I think maybe one problem is just the large variance in the length of these things. So that's
But I mean, there is this parameter, right? Coverage of the shorter sequence, 50% bland, and I mean, you get something. It's not like you will like do it perfectly correct. It's more just to get an intuition about the effect of the embedding-based clustering. Because after all, if this is really working well, then one could also consider creating protein data sets this way, right?
Yeah, that makes sense. Yeah, I think that would definitely be an interesting direction. Yeah, thanks.
Yeah, Denise, go ahead.
Thanks for a great talk. 2 questions. The first one is, how exactly do you concatenate 2 sequences together if you said that you are training with a fixed sequence length? Are you training like specifically for this task as well, or does your model generalize to extended sequences? And the second, inside this, adjacent to this is just, have you actually tried?
Let's first go with the first question and then with the second one.
Yeah, so yeah, that's correct. We use always a fixed length of 2048 tokens. And that's mainly a restriction of training efficiency. Yeah, like the ML models expect kind of a fixed length. There are kind of tricks you can play to increase the context length without retraining by like changing the position encoding parameters. We haven't really experimented much with that yet. But yeah, I guess another option is continually just increasing training in phases and continually increasing the context window.
Yeah, my question was actually focused on the last section you presented about protein-protein interactions, how you can combine 2 sequences, concatenate. You run the model on the concatenated sequence, right?
Yes, that's correct. We concatenate the 2 sequences and run it. Yeah, I guess maybe the second part of the question was for much larger proteins. Yeah, maybe it wouldn't fully fit within the context window. But for these,
and have you? Yeah, it's really interesting. The last part for me, if it is possible to predict known protein-protein interactions just from this like categorical Jacobian signal of diagonal.
Yeah, I think doing kind of a more a broader testing over broader ranges of proteins would be really interesting. Yeah, right now we've only done just a few, but yeah, expanding the set would be great.
Okay, thank you.
Okay. I don't. Yeah, if you have some other questions, raise your points. Otherwise, oh yeah, like so the reason why we train on these fixed size sequences is basically just because they are too long to fit into our context. So we chop them into stuff that fits into our context. Right. Okay. Good. Then thank you, Andre. I thought Jeffrey, go ahead. Last one for today. And Jeffrey's not unmuting. Good. Okay, go ahead.
You can hear me.
Yes.
I posted my question in the chat, but I just wanted to quickly ask whether you have any intuition on whether GLM2 would also be able to capture protein RNA interactions, because as you know, in the genome, you have coding sequences that encode, that get transcribed and translated into proteins, but you also have things that just get transcribed into mRNA or other non-coding RNA sequences that may interact with proteins. So I was wondering whether your model would be able to capture interaction between RNA and protein, essentially.
Yeah, that's a good question. We haven't.
I'm aware that you don't have results, but I just wanted to get some intuition.
Yeah, we haven't tested that, but I think that these kind of results might make us hopeful that that would work as well. Because we do in our context window during training, we do see, yeah, the proteins and the intergenic sequences. I think, yeah, hopefully we would also see some signal there. But yeah, we haven't tested it, but I think that would be a very exciting kind of direction.
All right, cool, thanks.
All right, then let us call it a day here. Thank you, Andre. And also, Andre, lots of, oh, I want this, oh, I want that. But of course, we are very grateful, and that's very cool what you've already done. That is to be sure. And always easier to criticize and say what's and to complain, but we also have to see how cool this stuff is, and I think we all appreciate it.
Yeah,
so
thank you
for this pretty cool work and this pretty cool talk. And yeah, I mean, we're also, I think, pretty impressed that by basically this slide that you're showing here, it really shows that there's something useful going on, right? Cool.
So, yeah, thanks so much, and thanks for the feedback. I think we'll definitely try to incorporate your ideas.
Very nice. Looking forward to what else Tata Bio cooks up. I vote for more philanthropy for, I don't know, yeah, philanthropy for science, and not just investing into startups and making startups do science or universities. So yeah, then see you, friends of the sun, around, and hopefully see you guys next week, and have a great start to the week. Bye, everyone.